Welcome back to Chapter 13, Bioenergetics and Biochemical Reaction Types. In this lecture, we will look at part 2 of this chapter. Here, we will continue our discussion on common biochemical reactions. We will then learn about phosphoryl group transfers and high-energy molecules, and then finish our discussion by looking at biological oxidation and reduction reactions. Group transfer reactions are inseparable part of biochemical pathways. Proton transfer is very common. As you may know, in enzyme active sites, proton transfer happens freely. Methyl transfer. Methyl transfer involves transfer of a methyl from a cofactor to a substrate, and this is usually catalyzed by enzymes. Methylation of proteins and DNA happens all the time. Acyl transfer. Acyl transfer reactions are pretty common, and one of the most common acyl transfer reactions happens during biosynthesis of fatty acids. Glycosyl transfer. This involves transfer of a sugar molecule, and it is usually catalyzed by enzymes. Phosphoryl transfer. Phosphoryl transfer usually happens and is catalyzed by enzymes called as kinases. And this is done on proteins and also on small molecules or metabolites. And this is usually done to activate metabolites and also is an important part of signal transduction. Substitution from the sp3 hybridized phosphorus in a phosphoryl transfer reaction proceeds via nucleophilic substitution, which is usually associative, SN2-like mechanism. The nucleophile forms a partial bond to the phosphorus center, giving a pentacovalent intermediate or a pentacoordinated transition state. And this is how it looks like. And if this were to be the center, electrophilic center, where a nucleophile, such as an alcohol, Z is equal ROH, attacks the phosphorus center, and if W is the leaving group, such as ADP, and what essentially happens is it goes via a pentacoordinated transition state. ATP is frequently the donor of the phosphate in the biosynthesis of phosphate esters, which involves phosphoryl group transfer. In this case, if you look at this reaction that is catalyzed by an enzyme wherein a glucose attacks this phosphoryl center of ATP, and this phosphate leaves from ATP to release ADP, and the phosphate is now stuck on glucose. This is a characteristic phosphoryl transfer reaction. Hydrolysis of ATP is highly favorable under standard conditions. That is because of 3 reasons. 1, there is better charge separation in products. Phosphate or ADP, either of them, there is much better charge separation. Second, better solvation of products. Phosphate or ADP are better solvated as compared to ATP. Third reason is because there is more favorable resonance stabilization of the products. As compared to ATP, the phosphate has more resonance forms as shown here, and ADP has more resonance forms than ATP. Now, the free energy change for ATP hydrolysis is minus 30.5 kilojoules per moles under standard conditions. But the actual free energy of hydrolysis, which is the delta G of ATP in living cells, is very different. And that depends on the cellular concentrations of ATP, ADP, phosphate, which is present as just phosphate. Magnesium in the cytosol binds to ATP and ADP, and for most enzymatic reactions that involve ATP as phosphoryl group donor, the true substrate is MgATP. The relevant standard free energy change is therefore that of MgATP hydrolysis to MgADP. Therefore, reiterating what I just said, the actual free energy change in a process depends on the standard free energy, the actual concentration of reactants and products. The free energy change is more favorable if the reactant's concentration exceeds its equilibrium concentration. The true reactant and the product are magnesium ATP and magnesium ADP respectively. The standard free energy change also is magnesium dependent. Hence, the actual free energy change is equal to a sum of standard free energy change plus RT ln concentration of magnesium ADP times phosphate divided by the concentration of magnesium ATP. Let us consider a problem here. From the data given in this table, let us calculate the delta G of ATP hydrolysis in rat hepatocytes at 37 degrees centigrade. Now, if we consider this equation, delta Gp, which is the phosphorylation potential, or the total free energy change of ATP hydrolysis, which is a combination of the standard free energy change and RT-ln concentrations of ADP, Pi, and ATP, which is shown in this expression. If you substitute for the standard free energy change, which is minus 30.5 kilojoules per mole, and at standard conditions, the universal gas constant and temperature, which is 37 degrees centigrade, and then substitute for the concentrations for rat hepatocyte, that is ATP is 3.38, ADP is 1.32, and phosphate is 4.8. And this is all in millimolar. And you would get a constant delta G value of minus 46.7 kilojoules per moles. So cellular ATP concentrations is usually far above equilibrium concentrations, making ATP a very potent source of chemical energy. Besides ATP, there are other phosphorylated compounds that have large delta G0 for hydrolysis. For example, phosphoenol pyruvate or PEP, the hydrolysis of which has a very large standard free energy change. Even larger than ATP, as you can see, it's minus 61.9 kilojoules per moles. And why is this? It is again because of electrostatic repulsion within the reactant molecule that gets relieved as a result of hydrolysis. The product, phosphate, is stabilized via resonance and gets solvated much more than the substrate or the reactant. The other product, pyruvate, that comes from phosphoenol pyruvate, undergoes tautomerization. Pyruvate as such is in enol form here, and it can go to keto form, and it can constantly interchange between these two forms. And these are the driving forces for the hydrolysis of phosphoenol pyruvate. The ATP hydrolysis reactions that we just saw is not just a simple hydrolysis. What I meant to say is, in this case, ATP doesn't just get hydrolyzed by water to ADP and Pi. what actually happens is ATP is involved in group transfer reactions. In this case, glutamate is converted to glutamine in presence of an enzyme, and ammonia is being used when this oxygen is replaced by NH2. Now, to facilitate this reaction from happening, coupling of ATP hydrolysis to ADP drives the reaction forward. So, throughout our next 3 chapters, we might see ATP being used in many different reactions and reactions that are written in one step. But in truth, or actually what is happening is a 2-step reaction. The first step is activation of this carboxylate by attacking ATP or attaching ATP and transferring a phosphate from ATP to this carboxylate. As a result, this carboxyphosphate compound forms, and then ammonia attacks this carbonyl carbon, which is now activated, thereby releasing phosphate, resulting in glutamine. So, in principle, ATP is facilitating a chemical reaction by making the substrate to have a better leaving group. And the thermodynamics that follows this is what drives the reaction. This graph here shows ranking of biological phosphate compounds by standard free energy of hydrolysis, as you can see on this axis. What this shows is the flow of phosphoryl groups represented by P and a circle from high energy phosphoryl group donors like phosphoenol pyruvate via ATP, that is shown here, to acceptor molecules such as glucose and glycerol to form their low energy phosphate derivatives such as glucose 6-phosphate or glycerol 3-phosphate. Now, the location of each compound's donor phosphate group along the scale is an approximate indication of the compound's standard free energy change of hydrolysis. Now, this flow of phosphoryl groups catalyzed by enzymes like kinases proceeds with an overall loss of free energy under intracellular conditions. Hydrolysis of low energy phosphate compounds such as glucose 6-phosphate or glycerol 3-phosphate releases phosphate, which has an even lower phosphoryl group transfer potential. Hydrolysis of thioesters is strongly favorable. For example, hydrolysis of acetyl-CoA is a favorable process. Acetyl-CoA, as you can see here, the structure, this is a CoA molecule, coenzyme A. We looked at the structure in the previous chapter, where it attaches, it has a thiol, and this thiol is usually the carrier of acetyl groups, and hence acetyl-CoA. Acetyl-CoA is an important donor of acyl groups, feeding 2 carbon units, just 1, 2, into metabolic pathways. And one of the most important functions is the synthesis of fatty acids. And when you look at the hydrolysis of acetyl-CoA, acetyl-CoA hydrolyzes to acetic acid, which then ionizes to form acetate, which is stabilized by resonance. The hydrolysis of acetyl-CoA gives a standard free energy of minus 31.4 kilojoules per moles. In acyl transfers, molecules other than water accept the acyl group, which means that in this case water doesn't accept, but other molecules accept the acyl group. Redox reactions, which involves oxidation and reduction, are another major source of energy for cells. Redox is simply electron shuffling. The forces that accompany the movement of electrons can be optimized to do work. Oxidation reactions are coupled to reduction reactions. This is an important slide because this gives you a pretty good idea as to how a reducing agent and an oxidizing agent works. Now, if X were to be a reducing agent and Y were to be an oxidizing agent, what happens is that electron flows from X to Y because X is the one that reduces Y. Anything that reduces sends or gives electrons to the one that gets oxidized. Now, the points are X loses electrons and X gets oxidized by Y, becomes more positive. The oxidizing agent, which gets reduced, gains electrons, and now it is reduced by X and becomes more negative because it's gaining electrons. Biological oxidations are often dehydrogenations because protons typically move along with electrons. Here are some examples of oxidation-reduction reactions. Reduced organic compounds serve as fuels from which electrons can be stripped off during oxidation. And that is exactly why fatty acids are a really good fuel, because they are highly reduced, right? And they are alkanes. So alkanes is the most reduced, whereas carbon dioxide is the most oxidized. And all these functional group containing molecules are in between. Carboxylic acid is more oxidized than aldehyde, which is more oxidized than alcohol, which is more oxidized than alkane. Biological systems use 4 mechanisms to transfer electrons. First, they can be directly transferred as electrons or as hydrogen atoms, as hydride ions, or through direct combination with oxygen. Reducing equivalents refer to the number of electrons transferred in a reaction, regardless of the method. Let us consider the reversible oxidation of a secondary alcohol to a ketone. Many biochemical oxidation-reduction reactions involve transfer of 2 electrons. In order to keep charges in balance, proton transfer often accompanies electron transfer. Now let's get to our example. In many dehydrogenases, these are enzymes that reduces molecules. The reaction proceeds by a stepwise transfer of protons and hydride. The enzyme lactate hydrogenase converts lactate to pyruvate, or the reverse, pyruvate to lactate. What it is doing when it is converting lactate to pyruvate is oxidizing lactate to pyruvate. Pyruvate is oxidized, as you can see it has a ketone, whereas lactate has an alcohol. The reverse process of converting pyruvate to lactate is a reduction. Now let us understand what reduction potential is. Reduction potential, E, is a measure of the affinity of the acceptor for electrons. This is similar to free energy. E0 is the standard reduction potential, and the units for this is volts. More positive E0 means more affinity for electrons. We calculate E via the Nernst equation, which is E equals E0 plus RT divided by NF, ln, concentration of acceptor divided by the concentration of donor. At 25 degrees Celsius, E is equal to E0 plus 0.026 volts divided by N, ln concentration of acceptor divided by the concentration of donor. Shown here is a table, and this table shows the standard reduction potentials of some biologically important half reactions. The reduction potential E essentially measures the affinity for electrons. A higher E, as shown here, means a higher affinity. Electrons transfer from lower to higher E, which means that electrons from this reactions can travel to this reaction. So this is the most reduced, and this is the most oxidized. And that's what this table means. Delta E relates to delta G. Delta E is given by E of the acceptor minus E of the donor. And delta G is directly related to delta E using this equation. Delta G is equal to minus NF delta E. And delta E is also given by minus delta G divided by NF, where N is the number of electrons and F is the Faraday constant. Now remember, delta G is equal to minus RT ln K equilibrium. So therefore delta E, the term for delta E becomes minus delta G divided by NF, which is equal to RT divided by NF ln K equilibrium. And hence we have the Nernst equation. Let us consider the conversion of pyruvate to lactate. What is this reaction? Is this an oxidation or reduction? A quick look at it says that pyruvate is getting reduced to lactate, right? And lactate is getting oxidized to pyruvate by the enzyme lactate dehydrogenase. So before we understand and calculate the reduction potential for the lactate to pyruvate reactions, let's look at the half reaction here. And the half reaction says that pyruvate going to lactate has a half an E0 of minus 0.185. Using this information, we can say that pyruvate going to lactate has an E0 of minus 0.185 volts, whereas lactate going to pyruvate, the reverse reaction, has an E0 of 0.185 volts. This is positive. So for the bolded reaction or the reduction reaction, pyruvate is reduced and is an electron acceptor in this case. For the reverse reaction, lactate going to pyruvate, lactate is oxidized and hence is an electron donor. Lactate and pyruvate needs a partner for a complete redox reaction because this is a redox half reaction. And that partner happens to be NAD+. Because in converting lactate to pyruvate, NAD+ has a very big role to play here. Because pyruvate, when reacted with NADH, gets converted to lactate and NAD+. So what's happening is NADH is acting as a reducing agent, and it gives electrons to pyruvate and gets converted to NAD+. At the same time, pyruvate, by accepting the electrons, get converted to lactate, and hence pyruvate is reduced and NADH is oxidized in the reaction. For the reverse reaction, lactate, when treated with NAD+, lactate gets oxidized and NAD, in this case, gets reduced. Now, if you look back at the table for the E0 value for conversion of NAD+ to NADH, it is minus 0.32 volts. And the reverse reaction is plus 0.32 volts. So for the bolded reaction, that is going from NADH to NAD+, NADH is oxidized and is an electron donor because NADH has electrons and it actually donates electrons as hydride. for the reverse reaction, NAD plus is reduced and an electron acceptor. So the individual half reactions are shown here. Pyruvate goes to lactate. The E0 value is minus 0.185 volts. At the same time, NAD plus goes to NADH, and the E0 value is 0.32. And pyruvate in this case is the electron acceptor and NADH is the electron donor. So how do you set this equation here? To calculate delta E is E0 acceptor minus E0 donor, or E0 reduction reaction minus E0 oxidation reaction. To make it less confusing, we'll just stick to E0 acceptor and donor, because what you have to do is to identify what is the electron donor and what is the electron acceptor. In this case, it's easy because NADH is the electron donor and pyruvate is the electron acceptor. So what we essentially do is we plug in the values, that is minus 0.185 of the acceptor minus 0.320 volts. That is equal to 0.135 volts. That is the delta E for these two half reactions combining to one full reaction. And using this value, delta E0, that is plus 0.135 volts, we plug into the Nernst equation to find out what the standard free energy change is, that is delta G0, that is equal to minus NF delta E0. And you can see that the standard free energy change is minus 26.05 kilojoules per moles. And this reaction is thus favorable. So NAD and NADP are common redox cofactors. These are commonly called pyridine nucleotides. They can dissociate from the enzyme after the reaction. In a typical biological oxidation reaction, hydride from alcohol is transferred to NAD+, giving NADH. So structurally, the difference between NAD and NADP is that in NAD, this is just a hydroxy, whereas in NAD+, this hydroxy is derivatized by a phosphate. But otherwise, the structure is very similar. You have the adenine, ribose, which is attached to phosphate, two phosphates, in fact, to another ribose that is attached to a nicotinamide ring. This is called as a nicotinamide, and hence the name nicotinamide adenine dinucleotide. Now, this is NAD plus because it's a positive charge, and this nitrogen that is shown here is an electron sink because hydride transfer happens at this carbon here. When hydride attacks this carbon, you can actually move this bond from here to here, and this bond can move here, and hence you get this kind of a structure. And this is NADH because this is the reduced form. Now the hydrogens, the reason why they are shown as a blue color or a pink color is because these hydrogens are on a prochiral carbon. Because if you replace one of these hydrogens by a different group, this carbon becomes chiral, right? So the hydrogen that is coming, the new hydrogen that is coming, can be on the top or on the bottom, depending on how the reaction happens. Formation of NADH can be monitored by UV spectrophotometry. We already know this, so we have done problems related to NADH absorbance. Measure the change of absorbance at 340 nanometers. This means that NADH, the reduced form of NAD, has a strong absorbance at 340 nanometers, whereas NAD+, which is the red colored band here, does not have any absorbance. Both NADH and NAD+ have absorbance at 260 nanometers. So this property of NADH can be used to study enzymes like NAD-dependent dehydrogenases. Another cofactor is Flavin adenine dinucleotide, or FAD. And FAD allows for single electron transfers. And it permits the use of molecular oxygen as an ultimate electron acceptor. There are flavin-dependent oxidases, which can oxidize various substrates. Flavin cofactors are tightly bound to proteins,